that don't know me, I'm Julian. I've been a research scientist and lecturer for about 40 years now. I'm not so active now. I took retirement a year ago, and but I'm still associated with the university as an honorary research fellow. So I still have a bit of work to do. In fact, one of my last PhD students just passed their exam last week, so I'm very happy about that. So what am I? Well, I'm a trained geneticist, and in particular, I'm a microbial geneticist. So I've spent my life looking at microbes of various descriptions. I did my PhD in virology, so you could say I'm basically a viral geneticist. Okay, but I have worked with other organisms. I've worked with the host cells of various viruses as well. What I'm not is an epidemiologist. Okay, so these are people that actually track the diseases around the world to see where they're breaking out and where they're not. And I'm not a clinical microbiologist. Okay, so I'm basically a geneticist who's interested is in how viruses and how other cells actually replicate and pass on their genetic information to others, including us. Right, so the talk of my, the subject of my talk today is COVID-19 and what you need to know. What I thought I'd do in this lecture is talk a little bit about some of the things that I think you should know about COVID, and to answer some questions that I've seen on social medias and other things where there have been some questions about what COVID and what the virus that causes COVID-19 actually is. So I shall go through a number of questions and talk about the science behind what the virus actually is and what it's doing. I think the questions I will be answering was, is COVID-19 flu? Why is COVID so nasty? Why is there variation in how people get affected by COVID? I shall then go and look at what the origins of COVID are, and then think about the idea, because I've seen it in some situations where people have said that it's a laboratory strain. So we will look at the evidences for whether it's a laboratory strain or not. And then we'll talk about vaccines, because we all want, I'm sure, this pandemic to end. We all want actually to get back on with our lives. So I will look at the basis behind the two main vaccines that we see them around today. And then I shall ask the last series of questions, which is grandpa safe? To look at the spread of the virus in Britain and around the world, and to look at the reasons for its transmission, which perhaps might help explain some of the government rules. So that's the sort of nutshell. Now, I aim to do this in about 45 minutes, but I'm here to enjoy myself. So if I get sidetracked or carried away, then Laura will actually put a little dinger on and tell me that I'm over time. But I think it's quite important that I leave a bit of time at the end for you to be able to ask some questions. Okay, now I'm sharing my desktop here. So I've got photo booth on so that you can see me talk. And beside of that, I've got my PowerPoint slides. I'm going to have to go backwards and forwards though. So now I'm on my PowerPoint slides, and you can see I've got a little laser point here where I can point to things. So the first thing that we really want to talk about is COVID the flu? And my answer to that is no, it is certainly not. The causative agent of flu are the influenza viruses, of which there are 3 types, A, B, and C. I put A up on my little table there. Whereas the causative agent of COVID-19 is a virus that's known as SARS-CoV-2. Okay. Now, these are very different viruses, although if you look at the pictures I put underneath there, they look quite similar. In fact, they're a similar size, but they're not the same. If we look at them from a classification point of view, then they're in the same kingdom because they're RNA viruses, but that's where the similarity ends. Okay, the differences between these two viruses starts at the phylum level. Now, if you want a comparison to what that means, if I was a zoologist and I was talking about differences between phyla, I'd be comparing an elephant with a snail. Okay, so that's, in terms of a virus, a virologist, that's the difference that we see between these two types of viruses. The differences are huge. The genomes are different. And although they have RNA, they have different types of RNA. The SARS-CoV-2 virus has what we call a positive strand RNA. And what does that mean? Well, that means that as soon as that RNA molecule enters a cell, it can be translated into protein and it can be replicated and packaged into viral particles. So it can get its replication cycle off really, really quickly. Okay. The big difference, though, between these two viruses is here. And can you see these little spikes that are poking out from these spherical shells? And these are known as the spike proteins. Now, the spike proteins of these types of viruses are really important because what they do is that they interact with the host cell that they're going to invade. So it's these little spikes that give the virus its specificity. Now, the spike proteins for influenza A infect cells that have cellularic acid and a specific type of cerulic acid. And that is actually found primarily in the bronchial tracts and in the lungs. So if you get an influenza infection, yes, you will have a lung infection, you will actually become ill, and you can develop pneumonia. Okay. SARS-CoV-2, on the other hand, has a spike protein that interacts with a different protein completely. Okay, and it's the interaction of this protein allowing the virus to get into the cell that makes it so nasty. So, why is SARS-CoV-2 so nasty? And why is it so variable? Well, what I've put up here is a sketch diagrams of the SARS-2 infection cycle, if you like, and its effects on the body. Okay. If we go down here, we can see a protein model of the spike protein that was found in SARS-CoV-2. What this is here is the receptor protein. Okay, so this is, if you like, a key and chain thing, okay, where this protein here, which is the receptor protein, which is normally found on the surface of the cell, binds into this groove here within the receptor protein, okay? And this receptor protein is known as ACE2. Now, ACE2 stands for angiotensin converting enzyme. And this is what actually is one of the reasons that makes this type of infection very nasty, because this receptor is an enzyme on the cell that converts the protein angiotensin. Okay, now angiotensin is involved in controlling whether your blood vessels dilate or constrict. Okay, now SARS-CoV-19 links onto this and gets engulfed into the cell. When it's engulfed into the cell, the proteins are stripped away, a process that we call unmasking, leaving the RNA molecules to be replicated and to be expressed into protein particles. Eventually, of course, these will form new viral particles, which will then be excluded from the cell. And as they excluded from the cell, they wrap themselves in the cell membrane. And then they go off ready to infect another cell nearby. So you can see that within this region, the virus can spread quite quickly to surrounding tissue. Now, the organ that the virus is targeting here is the alveoli. Okay, so these are deep in the lungs, and these are the if you like, the receptacles for oxygen exchange and carbon dioxide exchange with the blood. Okay, but the types of alveoli that the SARS infects is called type 2 alveoli. Okay, now type 2 alveoli are not involved in oxygen exchange at all. What they're involved in is actually producing a phospholipid that protects the lungs, okay? And they stop the lungs from collapsing. So if you have an infection where the virus is actually attacking the type 2 alveoli, eventually it's going to weaken the lungs and the lung will start to collapse. Okay, so as we go on, there's another thing that makes this type of infection quite nasty, because as soon as the virus starts to infect the lung tissue and the type 2 alveoli, what it does is that it starts to depress the expression of the ACE2 gene, so that the amount of the enzyme that's involved in converting one form of angiotensin into another form breaks down, which means that the form that actually is involved in vessel dilation, the opening up of blood vessels to allow blood through, to allow a higher oxygen concentration, is shut down. In actual fact, what you're left with is a type of angiostensin which is involved in constriction. So what we find now is that the lungs are the vessels are starting to constrict. So the vessels that are supplying the lungs can't get the blood in to get the oxygen around to the body. Now, of course, the body recognizes that this is happening. And what we're having now is what's known as cytotoxic damage. And cytotoxic damage releases an immunological response, which is basically producing what's known as cytokinins. Okay, these are basically interleukins, different types of interleukins that are produced. Now, interleukins, of course, are things that actually act against viruses. So they're doing the right thing. They're trying to produce these interleukins that are going to stimulate the immunity system to destroy the viruses. Okay. One of the things that they start to produce are these things called macrophages. And of course, macrophages are big cells, and they're basically, macro means big, phages means eaters, and they eat the tissue. Okay, so these start attacking blood cells, the blood capillaries, which then start to clot. So not only have we reducing the amount of oxygen that's coming into the lungs, we're actually then starting an immune response where we're actually blocking the capillaries up, which is once again more damage. And then we move into what's known as, and I'm sure you've heard of it, called the cytokine storm. This is where the body then goes and thinks, crikey, I'm really under attack here. I need to produce more interleukins, to produce more antibodies, to produce more phage cells. And of course, they will go off and they will then start attacking the damaged tissue, which will be the lungs. Now, at this stage, fluids were built up, and this is where you can get pneumonia. But the story doesn't end there. And the reason why the story doesn't end there is because this receptor, the ACE2 receptor, is not just present in the alveoli. It's actually present in other tissues. So we find it present in the kidney tissues. We find it present in the heart. We find it present in neurological tissues like the brain, as well as in the liver. And there are many other types of where it's found as well, including your throat. So what happens is that we have a reaction now against all these tissues that are being damaged. As the virus is being produced, it makes its way into the bloodstream, around the bodies. It starts then attacking these other cells. And this is when we have what's known as multiple organ dysfunction syndrome. And this is when the body starts to shut down. And of course, when that happens, you're actually close to death. So you can see really that this is not flu. This is something far more serious than flu. Okay, now why is it so variable? Why do some people have die from it? And now in this country, we're up to 60,000 deaths, which is a lot, far more than caused by flu. But some people seem to be unaffected by it. They seem to have it and they don't develop any of these symptoms at all. We know with flu that the people that are most susceptible to flu are the very elderly and the very young. But in this instance, what we're seeing is that the young don't seem to be so affected by it, whereas the elderly certainly are. Why is that? Well, the answer to that question is really to do with the distribution of the receptor protein, the ACE2 enzyme. In young people, it's not so prevalent in cells. So in actual fact, the density of these receptors is less. So even if they have a virus infection, it might not infect the cell. In which case, then the person isn't going to get ill at all. As you get older, you have more receptors around in the body, and of course, you become more susceptible to the COVID-19 syndrome. So now, that doesn't mean to say that young people aren't affected, because they are, and some people have died. In fact, one of the things that really frightened me when I first watched this in developing China was actually young people coming down with it, people in their 20s and 30s. And that scared me, because I expected young children, I expected the old. I didn't expect people in their 20s and 30s to come down with it. And that does happen. Okay, so I hope you can appreciate a little bit what's actually going on in the body now. What I'd like to do now is just sort of move on a little bit to talk about, there we go, the origins of the virus. And as we're probably all aware, I shan't dwell too long on this because I think probably most people are aware of this. The SARS-like viruses, like SARS-CoV-2, originated originally in bats. Why do we know that? Well, we've been studying bat populations for a long time. We know that there's a whole range of coronaviruses there, really a whole range. Why are they there in particular? Well, that's to do with the bat immunity system, which allows these viruses to replicate without actually damaging the bat very much. So we know that there's whole different types of coronaviruses. In fact, very early on, these types of viruses were highlighted as potential causes of pandemics because of this. Now, we've had two major SARS-like illnesses that have developed in the world. The first one was at the beginning of the noughties, and that was referred to as SARS, okay? And there, the origins of the virus were traced back to a bat, okay? But it wasn't the bat that transferred it to the humans, okay? The virus actually started to infect civets. Civets are mongoose-like creatures, okay? And the preponderance for catching civets and doing things with them actually lent it to spread into humans. SARS is very similar to SARS-CoV-2 in that it uses the same receptor, the ACE2 receptor, okay? We were lucky with SARS-2. SARS-CoV could have actually been as bad as SARS-CoV-2. The reason it wasn't was because the person that was really infected with this actually felt ill and went to a hospital. And actually, he was treated in the hospital. And luckily, the medics there, this is in Thailand, realized that there was a new disease. The guy actually recovered, but it was noticed that lots of people around the hospital started to pick up the disease. And in fact, some of those actually went to South Korea, where it really opened up and spread and killed about 8,000 people before the South Koreans got on top of it. So we were actually very lucky that because the initial outbreak was held in a hospital, that meant that it didn't spread around the world like SARS-CoV-2 did. The second example of this type of virus is known as the MERS virus. And once again, this was tracked back to bats, but the organism that it went through before it became infective in humans was the camel. And this virus was found primarily in the Middle East. Okay, this was actually quite a nasty virus, but once again, a vaccine was produced for that, which actually worked. And I'll talk about that a little bit more in a minute. Okay, so that brings us on to SARS-CoV-2. Where did it come from? Well, once again, the organisms the sequences that we look at are like those from bats. The organism that transferred it to humans, we think, is a pangolin, which is a scaly anteater. We're not too sure whether it was bat, pangolin, humans, or pangolin, bat, humans. That will become clear later on. But these viruses all attack the alveoli of the lungs, and they will lead to flu-like symptoms and develop worse than that. Okay, so having told you where the virus comes from, let's then have a look at, is it a lab construct? Because one of the things that I've seen floating around on the internet and all sorts of things is that this thing is a lab construct, that someone's made it in a laboratory and released it. Now, the ideas behind that have been either the most sinister, in that it was released specifically to collapse and destroy the Western civilization as we know it, to accidental release. So why do people think this? Well, the first reason is because these viruses are studied in laboratories, no doubt about it. And they're manipulated in laboratories, part of the public health concern. So you can't do this in any place. And where this research is done is in places like Portland Down, where they have very stringent control and conditions to handle these types of viruses. Basically, what we call, you need a category 4 laboratory to actually handle these safely. Which involves having negative pressures so that nothing escapes to the outside. Now, why have people thought that it's a lab construct? Well, there's two sorts of reasons that I'll go into. The first one was put forward by a French Nobel laureate that I shan't name because I think he must be hiding himself in shame at the moment. And he claimed that he detected a sequence in the SARS-CoV-2 genome that could only have come from laboratory manipulation. The reason for that is that he thought he detected a cloning, what we call a cloning vector. When we manipulate sequences in the lab, usually we put them into another molecule, usually a DNA molecule, so that we can manipulate it. And what this Nobel laureate claimed was that he detected part of this as a part of this sequence in the genome of SARS-CoV-2. Ipso facto, it must have been a laboratory construct. The only trouble with that is that the cloning vector that he was referring to was actually developed from a coronavirus. So in actual fact, it doesn't really hold water that this was a novel construct. The other reason why people have argued that, that this SARS-CoV-2 is a laboratory construct is because of something that's called a polybasic furin cleavage site. Okay, now if you look on the slide here, we can see that we have a bit of the amino acid sequence of the spike protein, okay, from different types of coronaviruses. And the bits we're interested in are these regions around here, okay? And these are cleavage sites. Now, for the virus to actually get inside the cell, the protein has to be cleaved, okay? Otherwise, it won't enter the cell. So these are really quite important sites. And these are the sites that are cleaved when the cell is actually entered. Now, here's SARS-CoV-2, and we can see that it has a cleavage site that is different to the other coronaviruses. Okay, and people have said, oh, it's different. That must have been due to a genetic manipulation. I don't actually believe that for one instance, because if you look at the sequence here for the MERS coronavirus, then we can see that actually these two sequences are very, very similar. And really the change that's happened here could well be explained as the virus has moved from one host to another. So perhaps going from the bat to the scaly anteater into the human, you might well expect there have been some evolutionary modifications that have allowed that change so that the virus can infect, say, the human cell more effectively. Okay. Certainly, if we run out sequences of the virus, we can get an evolutionary tree. And here I've put up an evolutionary tree. The coronaviruses are divided basically into alpha coronaviruses and beta coronaviruses. And we can see that all of the viruses that came from Wuhan in China actually formed a group with the beta, within the beta coronaviruses group. And these are traditionally affect bats. Okay. And in fact, the nearest thing we've got down here, the nearest ancestor, if you like, of the SARS-CoV-2 is SARS-CoV. Is SARS-CoV, the original SARS that was present years ago. Okay, so I don't think there's any debate in my mind that SARS-CoV-2 is natural. It's come from a bat population, probably infected another population and then got into humans. How we stop that, of course, is a different question. And certainly you might want to think twice about the importance of wet meat markets and things like that. But trying to get people out of these habits is a cultural thing. Okay, so I want to move on now to vaccine development and what we do with vaccine and what the two types of vaccines that you've heard about so far. Now, vaccines have been around for a long time, a long, long time. Certainly in the 18th century, vaccines became ideal, you know, a way of being protected from the work of Jenner and onwards. In fact, it was Queen Charlotte that actually made vaccination popular, because at that time, yes, there was even anti-vaxxers then who didn't believe in that, whereas Queen Charlotte actually decided to be vaccinated herself and had all the children vaccinated, which then actually made it very, very popular. When culture follows the royalty and things like that, so people became more vaccinated. Now, we've come a long way from actually taking a disease bit of related virus and then infecting people with that. And these days we have 5 main routes of developing a vaccine for a virus. Okay, I'm not going to go through all of these. Probably the ones that we know most about are the inactive vaccines, which is where we isolate the virus itself and inactivate it and then inject that in. We can have what's known as attenuating vaccines, okay, where we have the virus, but it's been genetically modified so it doesn't cause an infection. These are the standard ways of which vaccines work. And certainly when I was young and I had my polio vaccine, I certainly had one of these attenuated vaccines. But we've come a long way now. And the vaccines that are developed at the moment are really quite exciting. The ones that excite me the most are the nucleotide-based vaccines, okay? And this is the type of vaccine that Pfizer and Moderna have produced, okay? And we're going to talk about exactly what they are in a minute. The other type of development for SARS-CoV-2 has been a viral vector based on a vaccine candidate, okay? And so here, what has happened is that part of the virus, the SARS-CoV virus, has been put into another virus, which has been inactivated, and then that's been used as the vaccine. And this is the basis of the AstraZeneca, the Oxford AstraZeneca virus. So I'm going to talk about that first, and then I'll move on and talk about the nucleotide-based virus. Okay, so here we have a scheme for the AstraZeneca vaccine. Basically, what's been happened here is that the vaccinologists have taken another virus, in this case, an adenovirus. Now, adenoviruses are those viruses that causes colds, okay? But they haven't taken a human adenovirus, they've taken a chimpanzee adenovirus. I think I saw it advertised on Channel 4 News the other night as a monkey virus. Well, okay. I'm a biologist, and a chimpanzee is not a monkey; it's an ape, and it's our closest relative. Okay, so what's happened there is that we've taken the chimpanzee adenovirus, we've modified it so that it can't cause an infection, can't cause a cold, and then what we've done is we've taken the RNA from the SARS-CoV-2, we've converted it into DNA, and we've cut out the spike gene, or part of the spike gene, and splice that into the genome of the virus, so that when we grow that in the lab, the spike protein that's produced in this adenovirus is the SARS-CoV-2 spike protein. This then becomes the vaccine. That then gets injected into patients who then have an immunological reaction to it, but because their immunological reaction is going to be against the spike protein. This protein is a 2-dose vaccine. So you give the first dose to elicit immunological response, and then you give a second dose, it's called the booster dose, to increase the number of antibodies that are in the body, so that if a virus enters your body, then it gets destroyed. Okay. Now, this virus, I think there's been 2 studies that have been published so far about its efficacy. They say it's around about 90%, but that was from one study where they changed the initial dose that they gave. The World Health Organization says that a virus is effective if its efficacy is 70% or above. Okay, so the first study, its efficacy was about 62. The second study, it was about 90. So AstraZeneca now are running a more detailed clinical trials where they're going to boost that up. I'm pretty sure that this will meet easily the 70% efficacy level. Okay, why do I think that? Because this has been used before, and this has been used to actually develop a vaccine for MERS. And this little picture here shows the vaccine for the virus particle that's used as a vaccine for MERS. Okay, and that worked. In fact, that elicited a response within patients that lasted a year. Okay, which is good news. And I suspect this is the reason why AstraZeneca actually chose this route to go down, because it was a tried and tested way of actually developing a vaccine. The second vaccine that we hear about at the moment, which I find really exciting, is the Pfizer Moderna one. Okay, because that is actually so simple. Basically, what the scientists in Germany have done, in actual fact, has taken the gene that codes for part of the spike protein, okay, which is RNA, messenger RNA, and they splice that into another RNA molecule so that that bit can be expressed. They've then put that in a liposome, okay, and that's actually the vaccine. So that then gets injected into the cells. Now, there's one thing about membranes. Membranes have a tendency to fuse. So what will happen when that gets injected in is that the liposome will fuse with the cells. As it fuses with cells, that introduces the messenger RNA into the cell. The messenger RNA in the cytoplasm gets expressed to produce the spike protein for SARS-CoV-2, which then actually gets, because it's on a membrane, it gets exported to the outside of the cell. Now, your immunity system recognizes that and says, oh, this isn't me. I need to actually destroy that. So that will elicit an immunological response where T cells are produced and antibodies are produced. In actual fact, what will happen then is that the actual cell that's been infected actually gets destroyed. Okay, but it's elicited an immunological response. Now, this is a 2-dose vaccine, just like the AstraZeneca one. So the first dose elicits an immunological response. The second dose is the booster. So when the second dose is given, the same thing happens, but this time there are antibodies in there. That increases the number of antibodies and T cells that are around. That cell gets destroyed, but this time if any virus enters the cell, then it will get destroyed straight away by the antibodies. Now this is really quite neat. The ideas behind this have been around for about 10 years, and people have been experimenting with this for about 10 years. This is known as nanotechnology, if you like, and this is where people are worried about putting little motors in liposomes and getting them into cells and things like that. Well, yeah, sure. You might like that in science fiction. I don't think there's any real evidence of that actually happening at all. But what this really does steer the way for is for really effective vaccines against a whole range of viral diseases. This is really quite exciting. Okay, and of course, this is the one that our government, or our regulatory body, has approved for clinical use starting soon. There has been some criticism about that. Perhaps we can talk about that at the end. But as a virologist, I think this is really, really exciting and something that bodes well. Right, so we're moving into the last, if you give me 5 or 10 minutes more, Laura, I've been going on a little bit, but there's some things I think that are quite important that I want to say. And that's all to do is granddad safe? We know that nursing homes have been the hardest hit in this area. And really, if you're a public health scientist, the last thing you really want to do is to let a pandemic virus loose in your population. You don't want this thing replicating, because as it replicates, it mutates and it changes. Now, I know people have said, oh, well, the coronavirus, it doesn't mutate that much. If you let it actually, well, it's true, because it has a proofreading enzymes that do keep the replicated RNA down to a strict amount, but it still does mutate. And just to give you some idea about how it does that, for something that evolves, so it's not like HIV, which has a very high rate of mutation, not like that at all, but it still mutates and it still causes different lineages. At the moment, a Cambridge study has shown that there are 3 main types of SARS-CoV-2 in the world at the moment. In Britain, the sequencing analysis of the genomes of all the viruses really from the summer has detected 40,000 lineages. Okay, now this doesn't mean to say that they're separate. They all belong to one type of SARS-CoV-2 or another, but the reason why you have these differences is because they have mutational changes in them. And what is interesting, and what this map shows you, is that actually the viruses have been growing locally. So what we find is that we have different distributions of these lineages in different parts of the country. Okay, some of the lineages have become extinct, others have become more popular. Now, the worry about this is that if it's been transmitted locally, it really does stand a reason that the people in nursing homes at the moment, whereas before they were threatened from people coming from hospitals infecting them. Now it's actually coming from the community because it's so virulent within the population. Now, and once it's in a nursing home, it's really difficult to get rid of. It will work its way through it like a hot knife through butter, okay, which is not good. Okay. Now, just to give you an idea of the mutational changes that can take place, we'll move on to this slide here, which looks just at one mutational change. And this is a mutational change in the spike protein at amino acid residue position 614, where the residue has changed from a aspartate to a glycine. Okay. The one in China, the virus in China, had the aspartate at position 614. There was a mutation in Italy, okay, where glycine was replaced, okay, and that spread out. So this type, this lineage, isn't found in China at all. Okay, from Italy, this virus spread to France, from France and Italy into Germany and into the UK. Okay, now both Germany and the UK also had infections from the virus from China, okay, which in these diagrams here is the orange, the area is shaded in orange. The new virus from Italy is shaded in blue. Now, when people started to look at these different lineages in the laboratory, they started to get really, really worried, because the new 614G variant infected cells at a much higher rate than the original one. Okay, now the question then is, does this mean that it's going to be spread more easily? And is it going to affect the transmission rate more? So are we going to find this more than anything else? Okay, well, in Britain, we have been sequencing these things and we have been following them. Bit late, but we are doing it now. And these graphs here at the top of the page show the distribution of the two types of viruses, pre-March and post-March. And we can see that, yes, there has been a change, that there is more of the new variety around, but that only accounts for about 20%. So in actual fact, this is actually much lower than the rate at which we originally thought it might spread. So in actual fact, this mutational change doesn't look as if it's improving the transmission that much, which is good news. It could be a lot worse than that. But this is the sort of thing that happens when you let viruses spread unchecked. Okay, the last thing that I want to talk about in the last, promise you a couple of minutes, is the study that's been done in Iceland. And we don't hear much about Iceland, although I think they deserve a lot of credit, because in February, when they saw what was happening in China, the people and the government and the scientists in Iceland decided, well, we don't want to close our country down because we're really dependent on tourism. But we've got to do something to protect the population from this virus because it's likely to become a pandemic. So they decided very early on to actually use the standard methods, public health methods, of quarantine, tracking and tracing, and quarantining people. Okay, and that actually worked very well up until they've had a second spike now. But up until then, they had only 8 deaths per 100,000 people, whereas in America and here we're looking at 80 plus deaths per 100. Which is, you know, that's a whole magnitude of 10. So obviously people in Iceland have done something right. Okay, so they also, there was a director of a biotech company called Decode, and he went along to the Directorate of Health and says, I can help you here. They got all the universities in Iceland together, all actually sequencing and screening people. At the present moment, they've screened 55% of the population. Now, what they did was that they didn't just target severely ill people or mildly ill people, as we did in this country. They targeted everyone. And the horrifying thing that this data showed, because now they have a lot of data, first of all, was that in their initial studies, they were finding 30 to 40% of the people who were tested positive, and they didn't have any symptoms. So these were asymptomatic carriers that could transfer the virus. Okay, that's a frightening statistic. The other thing that they could do is actually track what the transmission is. And what this graph shows you here is the ratio of causes of transmission of viruses over October and November. Okay, and what we can see here is the ones that's really important are the household transmissions. These are the ones that actually are common throughout. Yes, we can see that there's rises in hospital infections, particularly as there's a rise in a peak, and then that goes into hospital and you see that go down, but then that comes down again. We can see that there's transmission at work, and we can see there's transmission at schools. But the one that really is important, the take-home message from that is it's household transmission that is actually causing the spread of the virus in Iceland. Now, this type of data, of course, is what the government's been looking at. And this is why they're going on so much about bubbles and limiting who you speak to. Okay, there's a website there. There's a new that just come out actually as an article in Nature about the Iceland approach. It's very laudable, and you should. Interestingly enough, the government and the scientists in Iceland wanted American scientists to join in, thinking that this would be a really good opportunity for American scientists to show what they're like, to show their mettle. But unfortunately, the federatory regulations prevented it. They stopped it dead. In this country, in the UK, we didn't make use of our universities at all. We have plenty of universities that have got grade 3 and grade 4 laboratories that could have done these testings. And most scientists would have been happily opened up their labs to do this. Okay. The government wanted to give it to people that had no experience in doing this at all. Commercial enterprises that have just been started up. Yeah, it speaks for itself. Right, now I'm going to finish now and open up for questions. There is one last thing I would draw your attention to, and that is an all-party parliamentary group that studied coronavirus and come up with some recommendations. And that's certainly worth reading.